So in this video, we're going to think about the structure and function of the plasma membrane, so the membrane around the outside of a cell. So we usually draw cells as a little diagram, it might be like this, and we've got the nucleus, we might have a mitochondria. What we're looking at is this outer membrane, the plasma membrane. Which is what's responsible for controlling transport in and out of the cell. So the inside of the cell we call the cytosol, and outside is just the extracellular. The extracellular environment. So that might be, pond water if it's a single-celled organism in a pond. So it might just be what's in the pond water, or maybe in the marine water environment. Or if it's a cell inside your body, that might be the fluids that surround all of our cells in our body that the cell might need to be able to respond to. So what we're thinking about is that plasma membrane around the outside of the cell. So what's the membrane made of? Well, we usually draw membranes that look something like this. So we've got, you'll often see this, as little cartoons of you sort of got a blob and two lines coming off it. Each one of those represents a molecule called a phospholipid. So what that molecule is there's sort of three components to it. So first of all, we have a central bit made up of 3 carbons called glycerol. And attached to those 3 carbon molecules, to 2 of them, we've got long chains which are called fatty acid tails. So those are basically made of carbons and hydrogens, and they have the property that they are hydrophobic, which means water hating. So you'll know, if you've doing the washing up or whatever, if you've got something fatty, then fats don't mix with water very well, so you kind of get fat droplets occurring. So these are fatty components. They don't like the water, they don't mix very well with water. So to 2 of the carbons we've got these fatty acid tails. To the other carbon, however, we've got a phosphate group, so that's a phosphorus and 3 oxygens. So that's your phosphate group. And that is a bit different because this bit of the molecule is hydrophilic, i.e. water loving. So this bit of the molecule likes the water, this bit of the molecule hates the water. So if I just colour code that on the cartoon, so the phosphate group and the glycerol are on the outside of the membrane, where they are able to directly interact with water molecules that we find both on the inside and the outside of the membrane. Whereas these bits in the middle, all these fatty acid tails, are in the middle of the membrane, so they're interacting with each other, and they are excluding water as much as possible. So that's the structure of the membrane. The membrane itself is pretty thin, so it's 9 nanometers thick. So that's really quite a small distance between let's put the external on this side and the cytosol on this side. Okay, so that's what the membrane's fundamentally made of. So we can continue that out. Lots and lots of lipids. These lipids can all move around, so they can they can move in that direction, and they can also flip from one side of the membrane to the other. But it's not just membranes in there. So one of the things that we have in there are little molecules of sterols, e.g. cholesterol. So you need cholesterol in your membrane, and stops the packing being too close at cold temperatures. So at cold temperatures, all of these lipids, they kind of get really sluggish and they kind of stick together and they get really gloopy. And if you have that, then the membrane doesn't work as well. You can't get stuff across the membrane. So those cholesterol molecules are in there every so often. They are on both sides of the membrane. In order to have the right fluidity of the membrane. So I'll just colour those cholesterols in pink. But they're found every so often through the membrane as well. So we've got phospholipids and we've got sterols. Then we've got some other components in there as well, because we also have proteins. So we have some proteins that are embedded in the membrane. So we stop having lipids for a little while and we've got what we call an intrinsic membrane protein. So that protein is physically stuck in the membrane. So let's colour that in, let's have yellow. So those proteins have got a variety of function. It could be an enzyme that happens to be in the plasma membrane, not in the cytosol. It could be some sort of transport protein to the molecules in or out of the cell. It could be a receptor, for example. So there are lots of different types of protein that sit and live permanently in the membrane. If they live in the membrane, and when you purify the membrane, they come with the membrane, they're called intrinsic. Those might have sugar residues, for example, on the outside of the cell. So that might be what we call a glycoprotein. And that can be quite important in recognition in the immune system, for example. So we might have sugars on the outside of the membrane. What we can also have are another class of proteins which are called peripheral membrane proteins. So let's have a peripheral membrane protein in red. So here there might be a little attachment, a little tail on the protein, but then the protein actually lives on the outside of the membrane. So this time it's on the cytosolic side. So that is a peripheral membrane protein. So if you purify the membrane, then usually peripheral membranes will come away and you'll find them in the cytosolic pure bit rather than in the membrane bit, because they're only quite loosely attached. They've got this little attachment there. So these are quite easy to detach from the membrane. Whereas these intrinsic proteins are really firmly embedded, so they tend to stay with the membranes. So once we've got these different types of system in the membrane, we can then start to think about how they might work. So what we might have, for example, so here's another intrinsic membrane protein, but this time it's got a domain on the outside. So let's colour that in yellow, because it's an intrinsic protein. So this might be a receptor. So you might have some sort of molecule. So you might have a signal that comes and binds to a receptor, which is an intrinsic membrane protein. That might then have a peripheral protein right next to it, and the intrinsic protein might activate the peripheral membrane protein. So let's call this protein A, this is protein B. So A, when the signal binds, activates B. B might then be able to move through the membrane as I say, we can have movement left and right in the membrane. That's okay, and that's actually really important for how the membrane functions. And then we might have another membrane protein. So again, this is an intrinsic protein. So our peripheral protein might move to activate protein C, and protein C might be a transport protein to let some new molecule inside the cell. So A is a receptor, B is a signalling protein, and C is a transporter. So we can start to see that these proteins interact with each other. They can move, particularly these peripheral membrane proteins are really good at moving along the membrane. And by the time you start to do that, then we start to have a signalling pathway. So now the cell is able to start organising itself so it can detect a signal using a couple of different membrane proteins, and then we have a molecule that moves in to the cytosols. So this is the inside of the cell, and then that might go, that molecule might go and then activate something else and something else. We might switch on genes, so we have a biological response. Not all signalling pathways involve the membrane, but the vast majority of them do. And we'll be thinking more about signalling pathways in some other videos later on in the module. So we've got phospholipids. So we've got all of these lipids here. They've got a hydrophobic and a hydrophilic side, which determines the overall structure. We've got sterols in the membrane that are there to make sure that it's got the right level of fluidity. And then we've got these intrinsic and peripheral membrane proteins that are stuck to the membrane, either very tightly or a little bit more loosely. They might be modified, particularly might have glycosylation on the outside, and they can start to be organised into signalling pathways that allow cells to respond to what's happening on the outside, so the extracellular environment. We might have signals that are then being perceived and having changes on the cytosol to give you a biological response.